So today we're going to talk about the production of eggs and sperm from germ cells. This is a little different than mitosis that we just learned about. This is meiosis. In meiosis, a process of reduction division in which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in a diploid cell. A diploid cell is one in which there are two sets of chromosomes, one from the father and one from the mother. Haploid is a term that means there is one set of chromosomes, such as we find in sperm and egg cells, which join to make a 2N zygote. Homologous chromosomes that each have a corresponding chromosome from one from mother and one from father, from each parent. Meiosis involves two distinct stages, Meiosis I and Meiosis II. I often call them mitosis, because in the first stage, we get two daughter cells that are both 2N. They have both complements of a complete complement of all their DNA. But the difference between meiosis and mitosis is that crossing over occurs in meiosis I, so that genetic material is exchanged between the chromosomes from mom and dad, so that you become a unique individual. In meiosis II, the two daughter cells created by meiosis I divide, but the DNA is not replicated. So when it divides between the two daughter cells, we get 4 daughter cells because each of the cells produced from meiosis I produces, there are 2 cells there. So each of these then goes through a reduction division, so we end up with 4 cells. And each of them has only half of the genetic material. Each chromosome pairs with its corresponding homologous chromosome to form a tetrad. So we now have 4N in the center of the nucleus. Briefly, crossing over may occur here. Crossing over is when chromosomes overlap and exchange portions of their chromatids.
The earliest part of the first stage of meiosis, called Prophase I, begins just as it does in mitosis. Chromosomes form from chromatin, and these chromosomes contain DNA replicated during the preceding interphase. As Prophase I progresses, 2 things happen in the germ cells that didn't occur during prophase in mitosis. First, the identical or homologous chromosomes form pairs with one another, so that the tall chromosomes find their mates, and the short chromosomes do likewise. This pairing of homologous chromosomes during the first prophase of meiosis is defined as synapsis. The second difference between prophase I of meiosis and the one we saw in mitosis is that parts of one paired chromosome can actually be exchanged with parts of its opposite pair. This process, called crossing over, is defined as the exchange of chromosomal material during synapsis and occurs only during the first prophase of meiosis.
Then, in metaphase I, spindle fibers attach to the chromosomes.
During the next stage of meiosis, called metaphase I, the centromeres of the homologous chromosomes line up on the equator of the cell. In contrast, during mitosis, the centromeres of the homologous chromosomes align themselves in a completely independent fashion.
Anaphase I, now is when they pull apart.
During the third stage of meiosis, called Anaphase I, the homologous chromosomes, each made up of 2 sister chromatids, move apart, centromeres intact, one toward each mitotic center. In contrast, during mitosis, the centromeres separate during anaphase, and the sister chromatids move apart as new daughter chromosomes.
So we see that each cell that's going to form is going to be a 2N cell with a complete complement of DNA. Then in telophase I and cytokinesis happens, the nuclear membranes form and the cell separates into two cells.
During the fourth stage of meiosis, called telophase I, each new cell contains just one of each homologous chromosome. And each of these chromosomes still consists of two sister chromatids.
Well, we get two haploid cells. Each cell has half the number of chromosomes as the original cell in prophase II.
The fifth stage of meiosis, called Prophase II, follows right on the heels of Telophase I, without an interphase stage in between them, as happens in mitosis. This means the DNA of the chromosomes in the Prophase II cells has not been replicated. And this explains why the chromosomes of Prophase II cells have not sprouted any new sister chromatids. From now on out, meiosis follows the same stages as mitosis.
So the chromosomes line up, similar to metaphase in mitosis, and they're going to be pulled apart by the spindle fibers.
During the sixth stage of meiosis, called Metaphase II, the chromosomes line up on the equator of the cell. And now the centromeres split apart.
So the sister chromatids separate and move to the opposite ends of the cell, and we get Anaphase II, where they're being pulled apart to the opposite ends of the cell.
And during the seventh stage of meiosis, called Anaphase II, the centromeres separate for the first time. And now the sister chromatids become daughter chromosomes as they move toward the opposite poles of the cell.
So meiosis II results in 4 haploid cells. The last stage is telophase II.
Finally, during the last stage of meiosis, called telophase II, new nuclear membranes form. And as the chromosomes change back into chromatin, we can see that 4 new sex cells have been produced from the single original diploid germ cell. But since the DNA was only replicated once throughout a series of 2 cytoplasmic divisions, the final number of chromosomes in the sex cells is only half the number present in the diploid parent cell. In other words, the sex cells are haploid. Now, when 2 haploid sex cells, sperm and egg, join together, the fertilized egg will contain the same diploid number of chromosomes found in any body cell.
So in males, meiosis results in 4 sperm cells, but in females, meiosis results in only 1 egg and 3 polar bodies, which are not used in reproduction. The eggs are all produced in human females while they are still babies in their mother's womb, and they don't mature until puberty. So a comparison of meiosis and mitosis is, mitosis results in 2 diploid cells. Meiosis results in 4 haploid cells. Cells are genetically identical in mitosis, but in meiosis, cells are genetically different. Mitosis occurs in the somatic cells, the body cells, and meiosis occurs in sex cells, otherwise known as germ cells, associated with reproductive organs.